Good morning, everybody. Welcome back to Daily Lectures by New Age Cosmetic White EdTech Healthcare Consultancy. I'm Dr. Bobby back with you. We're on the final sections of the subject of molecular biotechnology on Chapter 23, titled Societal Issues in Biotechnology. The specific topics covered under this section are, concerns about the safety of consuming genetically modified foods, alteration of the nutritional content of food, potential for introducing toxins or allergens into food, potential for transferring transgenes from food to humans or intestinal microorganisms, controversy about the labeling of genetically modified foods, concerns about the impact of genetically modified organisms on the environment, impact of biodiversity, impact of the Bt toxin on non-target insects, environment benefits of genetically modified organisms, economic issues, who benefits from molecular biotechnology, how the views about genetically engineered food impact trade, summary, and review questions. Coming back to the matter proper. No technology is without risks. However, it is important to weigh the benefits of a technology development against the risks and to manage the risks in a responsible and informed manner. And to manage the risks in a responsible and informed manner. Over the last 30 years, recombinant DNA technology has provided many commercial products described throughout this book that benefit society, and after much scientific scrutiny, most have proven to be safe. For the most part, society has accepted the technology vaccines and other medicines developed using recombinant DNA technology are generally accepted by the public and considered to be necessary and at least as safe as recombinant medicines. On the other hand, food products derived from transgenic plants and animals make some people uneasy. They question whether the introduction of a transgene may make the food toxic, allergenic, or less nutritious, or whether the transgenes can be transferred to other gut organisms, or perhaps even to the consumer during digestion. Furthermore, some worry that once a transgene is released from the laboratory environment in a cultivated plant or a farmed animal, it will have unintended harmful consequences that cannot be foreseen or controlled. Some opponents of molecular biotechnology will never be convinced that the products are safe. For them, the transfer of genes among unrelated organisms is unnatural and therefore inherently wrong. Others want assurance that the risks are minimal and are justified because the products are necessary and that society in general, rather than a few select interest groups, will benefit from their availability. Concerns about the safety of consuming genetically modified foods. All foods produced today are derived from plants and animals that have been genetically manipulated to enhance desirable characteristics through either selective breeding or recombinant DNA technology. The genetic changes introduced through recombinant DNA technology are vastly smaller and better understood than those from traditional breeding, as only a few well-characterized genes are introduced. Also because known regulatory elements are included on the introduced genetic constructs, the spatial and temporal expression of recombinant genes can be controlled. Despite these advantages, some consumers are concerned that manipulating the genomes of food plants and animals through recombinant DNA technology can lead to the production of foods that are unsafe for human consumption. This fear has had a powerful effect on the food industry. For example, consumer concern over the safety of transgenic foods led major baby food producers to stop using them in their products, and several fast food chains have moved them from their menus. On the other hand, proponents of genetic engineering argue that by rejecting transgenic ingredients, producers are marketing foods that are less safe. Conventional food is more likely to be contaminated with potent mycotoxins produced by molds, which could be prevented by growing crops that are engineered to be mold resistant. Allergens, naturally present in many foods, could be removed through genetic manipulation. Alteration of the nutritional content of food. All genetically engineered foods derived from plants and animals are tested on a case-by-case basis for substantial equivalence before approval for commercialization by regulatory agencies. This means that the safety and nutritional content of genetically engineered food must be substantiated. Substantially the same as those of the corresponding conventional food. The nutrients that are measured include carbohydrates, proteins, fats, amino acids, fatty acids, vitamins, and minerals. The natural variation in nutritional content among the conventional varieties of the food product due to varietal, developmental, or environmental factor is taken into consideration, and therefore there is usually a range of levels that are considered acceptable for a given food. Also considered are any changes in any anti-nutritional factors that occur naturally in foods. These include chelators such as fatty acid, which effectively remove calcium, iron, zinc, and magnesium, protease inhibitors, which prevent protein digestion, and lectins, which reduce the bioavailability of carbohydrates. The measurements are used to determine whether undesirable changes in nutrient content have occurred as a consequence of genetic manipulation, that is, whether levels of important nutrients are reduced or levels of potentially harmful compounds have increased. Numerous feeding trials in which insect-resistant or herbicide-tolerant transgenic corn, maize, rice, potatoes, soybeans, or tomatoes were fed to laboratory animals or livestock for prolonged periods, and often for several generations, have found no adverse effects related to nutrient deficiencies. There were no significant differences in the composition, quality, or digestibility of the food, or in the development, health, or performance of animals fed genetically engineered and conventional plants. For example, more than 20 studies have shown that the yield and quality, fat, lactose, and protein content of milk produced by lactating dairy cows fed corn genetically engineered to be insect-resistant, or corn, soybeans, or beets genetically modified to be tolerant to the herbicide glyphosate, are the same as those of milk from cows fed on a transgenic diet. Detailed analysis of the compositions of products derived from cloned animals has led the U.S. Food and Drug Administration Center for Veterinary Medicine to conclude that there is no difference between the nutritional element contents of meat or milk from clones and conventionally bred animals. These studies measured milk yields and milk and meat fat, protein and carbohydrate contents. The amounts of amino acids, fatty acids, and important vitamins and minerals were also determined. It is acknowledged that an exhaustive analysis of tissue composition is impossible due to the complexity of the molecules present, and that composition is impacted appreciably by the diet of the animal and its environment. Several animals have been engineered to improve the nutritional value of food in these cases. The nutritional content of the genetically engineered food is intentionally made different from that of the conventionally bred plant, and in this regard does not meet the criteria for substantial equivalence. An important example of a nutritionally enhanced food is golden rice, which was created to address dietary deficiencies of vitamin A. Insufficient consumption of vitamin A is a significant problem in developing nations where each year hundreds of thousands of children are blinded due to retinal and corneal damage, suffer from infectious disease, or die as a consequence of this nutritional deficiency. Carrots, tomatoes, meat, and milk are good dietary sources of vitamin A. However, in developing countries, they are not always readily available. Rather, rice grains, which do not contain vitamin A, are a primary food source. In 2000, after 7 years of research, scientists successfully engineered rice to produce beta-carotene, a biosynthetic precursor of vitamin A, by introducing genes from daffodil and the bacterium Ovenia urivora. In the initial strain, the levels of beta-carotene were too low to provide the recommended amounts through diet, a problem highlighted by opponents of the genetically engineered rice. However, a subsequent version, announced in 2004, introduced 20 times more beta-carotene, thereby reducing the amount of rice that would need to be eaten in order to obtain sufficient vitamin A to stave off malnutrition. Despite the promise of this nutritionally enhanced food to solve vitamin A nutrition, almost a decade after it was first developed, golden rice is not yet available commercially. Among other extensive environmental and biosafety analysis, golden rice, as for other nutritionally enhanced foods, must be assessed for substantial equivalence in nutritional composition to non-transgenic rice, except for the compound that is intentionally altered, that is beta-carotene. Where all of the compositional qualities are similar to those of a non-engineered comparator plant, the safety of the altered nutrient levels is evaluated against well-established nutritional guidelines. The large amounts of golden rice required for a completely safe assessment have been difficult to obtain in the greenhouse and in the limited field trials approved so far. Lack of financial support for a product that is being developed for low-income consumers and aggressive opposition to nutritionally enhanced food developed through genetic engineering have delayed completion of the requirements of regulatory approval. Opponents argue that rice containing the vitamin A precursor is unnecessarily risky because other methods to deliver vitamin A are available, including vitamin tablets and vitamin-fortified foods such as sugar and growing vegetables rich in vitamin A. However, these methods are often costly and difficult to sustain. Despite the obstacles, holders of the various patented technologies that contributed to the development of golden rice provided licenses to developing countries at no charge in anticipation of its eventual commercialization, expected in 2011, as a humanitarian gesture to improve the health of billions of people. Potential for introducing toxins or allergens into food. It is important to bear in mind that no foods can be guaranteed to be 100% safe. Many foods contain trace amounts of natural toxins that are harmless when consumed at low levels. For example, two common food plants, the potato and tomato, are members of the nightshade Solanaceae family that produce glycoalkaloids that can cause serious illness when consumed in large or relatively large quantities. Also, many foods naturally contain proteins that elicit an allergic response in some consumers. Extensive health safety tests of animals that compare genetically engineered foods to their non-engineered counterparts have concluded that the process of creating genetically engineered foods does not make the food different in digestibility or detrimental to the health of the consuming animal. However, it is necessary to ensure that the introduction of a specific transgene into a food product does not increase the risk of producing a toxin or allergen. The regulatory agencies consider each food product derived from a transgenic plant or animal on a case-by-case basis and assess the risks against those associated with consuming the corresponding non-transgenic product. There have been some highly publicized reports of adverse effects that have propagated unease among some consumers about eating genetically engineered foods. Often the reports in the popular press have focused on particular aspects of the story that have led to confusion or have failed to mention that the data do not hold up to scientific scrutiny. For example, in August 1998, ARPA birds die The scientists at the Rover Research Institute in Aberdeen, Scotland, announced on a British television program that rats fed transgenic potatoes over 110 days were stunted and had suppressed immune function. He did not mention that the transgene encoded a plant lectin, a carbohydrate binding protein. He also failed to emphasize that the experiments were preliminary and were being conducted to determine if this particular genetically modified plant was actually safe. Putztai's revelation became instant news, with the focus of attention on transgenic plants in general and not the specific gene that have been introduced into potatoes in this instance. The incident was further complicated when Putztai's data were found by the director of the institute to be deficient scientifically. Eventually, after much controversy, Putztai and an associate published a study suggesting that rats fed a diet consisting solely of transgenic potatoes, engineered to express a plant lectin gene, had compromised immune systems. Independent analysis of this report by a committee of the Royal Society in Britain and the National Institute for Quality Control of Agricultural Products in the Netherlands found serious scientific shortcomings that brought into question the validity of the results. Despite these experts' assessments, this transgenic potato saga was used by the critics of biotechnology as proof. Despite these expert assessments, this transgenic potato saga was used by the critics of biotechnology as proof that transgenic plants are inherently dangerous to humans. A transgene that is commonly introduced into crops encodes a crystal cry protein, an insecticidal toxin from the bacterium Bacillus thuringiensis Bt toxin. When ingested by a target insect larvae, the toxin interacts with the specific receptors in the epithelium of the larval gut. The protein inserts into the epithelium, forming a pore through which gut contents leak, leading to the death of the insect. The Bt toxin has been safely used for over 40 years to protect crops from a variety of pests, either as an agricultural spray or produced by transgenic plants. The cry protein is produced by beta thuringiensis in an inactive form as a full-length protein. Proteases produced specifically by the insect cleave the protein, which activates the toxin. The proteases are active in the slightly alkaline environment of the insect gut. The protein is safe for human consumption because humans do not produce the specific protease required to activate the toxin or the epithelial receptors that bind the toxin, and the toxin is rapidly degraded under the acidic conditions found in the mammalian digestive system. Nonetheless, some consumers have questioned whether the cry protein may be a potential allergen. This concern is fueled in part by reports that are often based on misinterpreted scientific studies. For example, in a 2005 campaign against Speedy rice, Greenpeace stated that the rice genetically engineered to produce the cry A1AC protein could cause an allergic reaction as it did when tested on mice. Several scientific studies were cited to support the claim that the protein elicited an allergic reaction in mice. However, it was not explained that the purpose of the cited studies was to test the cry1AC protein as an adjuvant to increase the efficiency or the efficacy of a vaccine, or that the cry1AC protein was chosen because it was known to have low toxicity in vertebrates. While anti-cry1AC antibodies typical of an immune response were produced in the immunized mice, they were not immunoglobulin E antibodies involved in allergic response, nor was evidence of an allergic reaction presented. It was also not mentioned that cry1AC was already widely produced in crops consumed by humans and other animals without harmful effects. Interestingly, there have been almost no objection to the spraying of the Bt toxin over the last 4 decades. Opposition seems only to be voiced against its production in transgenic plants. To further support the allergenicity of cry1AC, opponents made reference to concerns among regulators regarding the potential allergenicity of another BT product known as Starlink corn, an unfortunate association due to a regulatory breach by Starlink. The beta-thyrogenesis gene used to generate Starlink corn encodes the KORAH-9C protein, which differs from KORAH-1HC and other BT toxins. Although KORAH-9C is not similar to known protein allergens and is not derived from an organism known to produce allergenic proteins, it has greater heat stability and is not as readily digested, which are characteristics of some allergenic proteins. For these reasons, the FDA did not approve Starlink for human use, but did allow it to be sold as animal feed and used for industrial purposes. However, in July 2000, Larry Boland, director of the Community Health and Environment Program for the U.S. affiliate of the International Environmental Organization Friends of the Earth, went grocery shopping and brought a large number of products that contained corn flour, including taco shells, cornflakes, and muffin mixes. He had these tested for the Starlink KORAH-9C gene using the polymerase chain reaction test, and the evidence of the gene was found in some of the taco shells. Clearly, food safety regulations had been broken. It was estimated that Starlink contaminated at most 0.125% of all corn-based foods. The source of the contamination has never been traced. Ventus, the company that developed Starlink, has assumed that some farmers probably inadvertently mixed it with other varieties of corn despite written agreements that committed them to keep their harvested BT crops segregated. The seed is no longer sold. The Starlink episode had a number of ramifications. The critics of biotechnology pointed out that the regulatory system was seriously flawed and stressed that consumers should be aware of the hazards of genetically modified organisms. The biotechnology industry representatives noted that there was no public health concern, but that the regulatory issue had to be corrected. The FDA declared that it would no longer allow a product to be approved only for animal feed. A number of scientific advisory panels were convened to study in detail the consequences of KORAH-9C in human food, the possibility of consumer maladies due to eating products that may have been derived from Starlink, and how to improve the regulation and surveillance of transgenic crops. By 2001, there were no different definitive cases of allergenic reactions by KORAH-9C, but individuals who had symptoms compatible with a possibly newly acquired allergy was monitored. No serious illness has been documented as a result of eating food contaminated with Starlink. The potential for introduction of allergenic proteins into a food product that does not otherwise elicit an allergic response is a major concern. The FDA requires labeling to indicate genetically engineered foods containing proteins from organisms known to produce proteins that are allergens to humans. Many major allergens and proteins from eggs, milk, shellfish, fish, tree nuts, soybeans, wheat, and peanuts. In most cases, such transgenic food products are not commercialized. For example, soybeans engineered to express a protein from Brazil nuts that was intended to increase the methionine content of the soybeans were found to react with syrup from individuals known to be allergic to Brazil nuts. As a consequence, further development of the transgenic soybeans was voluntarily terminated by the developer. Food allergens are a significant problem in developed countries. In the United States, about 5 to 8% of children and 1 to 2% of adults have food allergies, some of which can be fatal. Molecular biotechnology has the potential to reduce the allergenicity of some foods by preventing the synthesis of the allergenic protein. A hypoallergenic variety of peanut is currently under development using RNA interference to reduce levels of the seed storage protein AIIMS-NEET, the most potent peanut allergen to which most hypersensitive individuals respond, often fatally. Potential for transferring transgenes from food to humans or intestinal microorganisms. We consume a large amount of DNA and the encoded protein products in the plant and animal tissues that make up our daily diet. Indeed, these supply part of the nutrition from food. It has been estimated that an average adult consumes approximately 0.1 to 1 gram of DNA per day. The DNA is partially degraded during food processing at high temperatures or low pH and is further degraded into small fragments through chewing and the activities of nucleases in saliva and the gastrointestinal tract. Only a very small amount of the total DNA consumed remains in fragments that are capable of carrying an intact gene as the digested DNA passes into the small intestine. Excuse me. This was shown in patients who had an ileostomy, an operation in which the upper portion of the intestinal tract, the ileum, is severed from the lower portion of the small intestine, which allowed researchers to collect digestion products before they completed passage through the small intestine and colon. After eating soybeans genetically engineered with EPSPS gene encoding 5-enol pyruvyl shikimate 3 phosphate synthase, which confers tolerance of the herbicide glyphosate up to 3.7% of the consumed transgene, was detected by PCR as a 180 base pair fragment in the digestion products of the ileostomist. The full-length EPSPS gene, 2.27 kilobase pairs, was detected in many of the collected samples. However, the EPSPS gene fragments were not detected in the feces of healthy volunteers with intact intestinal tracts and therefore were completely digested in the small and large intestines. If the genes that are present in the food we eat remain intact in our intestinal tracts, can they be incorporated into genomes where they could disrupt the function of a gene or where they could be expressed, resulting in the production of a foreign protein? Several studies have shown that neither transgenic DNA nor the recombinant proteins encoded by the transgenes present in food are found in the tissues of humans and livestock that consume the food. PCR and southern hybridization failed to detect recombinant DNA in the milk, eggs, skin, muscle, and other tissues of several livestock, animal-fed, genetically engineered crops. Non-recombinant chloroplast DNA, which is naturally present in multiple copies in plants, was detected in some tissues. This may have implications for transgenes that are incorporated into the chloroplast genome. Even present in tissues, integration of functional genes from fruit DNA, transgenic or otherwise, into the genome of the consuming animal has never been found and is likely restricted by 1. the small fragments sizes of the DNA remaining in the gastrointestinal tract following food digestion. 2. The presence of cytosolic nucleases that further digest foreign DNA that is taken up by intestinal epithelial cells. 3. Insufficient sequence homology for integration by homologous recombination. And 4. The requirement for appropriate transcription and translation signals for expression of an integrated gene. The possibility that antibiotic resistance genes used as selective markers during the process of creating transgenic plants could be transferred to microorganisms in the intestinal tract has also been investigated. Since most of the DNA derived from food that reaches the intestines, and particularly the colon, where most gut microorganisms reside, has been digested into small fragments, the risk of an intestinal microbe taking up and expressing a functional antibiotic resistance gene is extremely low. For example, an antibiotic resistance gene in transgenic corn was undetectable after 1 minute in sheep rumen fluid and the intestines of chickens, although there is no evidence from long-term studies of a variety of animals that antibiotic resistance genes consumed and transgenic food have been transferred to intestinal bacteria to alleviate public concerns. Alternate selection methods that avoid antibiotic resistance genes have been developed. Controversy about the labeling of genetically modified foods. The labeling of biotechnology-derived foods is a contentious issue. In the United States, the composition of the products, not the process by which the product is produced, determines whether specific information should be added to a label. This lecture ends in a couple of seconds from now. The rest of the portions will be continued in the next lecture. Please take notes and screenshots if any. Kindly do not commercialize any aspects. Use them to the best of your studies for a good career. Best wishes. Thank you.